record anything popped up?
Yeah.
But cannot. Okay. What's the structure of DNA? What's the structure of DNA?
The base.
Okay. Okay, so did I talk at least a little bit about the cell cycle? Yeah, on page 84, the cell cycle. Okay, so maybe just put things in context where we are for this particular lesson. Yeah, then you won't see out of the blue why we're doing this. Okay, so quick recap. The cell cycle. What we learned is that cells don't live forever. If cells don't live forever, they only go through a cycle of themselves, by themselves. Each cell goes through a cycle of growth. It also divides. If we look at the cell cycle, a very small proportion of it is used for the process of cell division. This is when the cell divides. But it spends most of its cell cycle in a very large phase we call interphase. We call it interphase because it's like intermission. It's like the phase it spends most of its time in, and then here's where the main big action really happens. Under interphase, we can break it up into three more portions. Looking at your textbook, and maybe from what we talked about last round, there are several parts to it. There's G1, and then there's S, and then there's G2. The G stands for growth. So in these segments is when a lot of growth takes place. This is when I would grow and produce a little bit more organelles, more cytoplasm, more cell membrane. S is the phase that we call the synthesis phase. This is the phase where we synthesize something also. What do we synthesize in this phase? Specifically, DNA. DNA is what we synthesize here. More specifically, I'm going to use a new word today. We're going to do DNA replication. The idea of replication is to make another copy of. That's what DNA replication is. And it takes place only in the S phase. So the cell actually goes through a period of growth, then I replicate my DNA, and then I grow some more. And then once everything is ready, then it starts to divide. I have a question for you. Why must it grow? Why must it produce double the amount of DNA before it divides? What if I just skip out the interphase and just divide?
You end up with nothing in the end.
I'll end up with nothing in the end. What do you mean by that?
If you divide, you have to give half the DNA, right?
Okay, if I divide, I have to give half of the DNA to both.
And you keep half the DNA.
And I keep halving it, and I keep halving it. Actually, if I keep doing that, each cell will become smaller and smaller and smaller. Each cell will get less and less and less DNA. That's what you mean by nothing in the end. I don't know what you end up looking like, but you get the point. It kind of becomes smaller and smaller into a cell that's no longer going to be effective for use. That's why the cell prepares itself before it divides. It grows, it produces double the amount of DNA before it goes into the division. Interphase, our focus is really the S phase. G1, G2, for your information. By G, the G itself reminds you is for growth. But we are really only going to focus on the S phase. This is the exciting and interesting part that we're going to focus on. For whatever is left for the mid-years, actually this is what's left up. We're going to learn about the synthesis phase. Under synthesis, we are going to learn about how DNA replicates. And also we're going to learn about an experiment about how we found out how DNA replicates. Two parts to it. So this is part one today of the story. Next week we're going to go into part two, asynthesis. The next part that we're going to cover is we're going to go into the cell division part. We're going to go into mitosis and cytokinesis. This is the second part of our learning. This part is a bit shorter. You just need to know the stages of mitosis and cytokinesis. So this is what we are left with for the mid-years. Okay? If you have been consistent in your revision, you can take comfort in the fact that you have one more chapter stack to cover these three things. Okay? I hope you feel a little bit comforted if you have been consistent now. Okay? So this is what we're left with. Today, our focus is just here. DNA replication. We want to find out how DNA replicates. For those who have done their reading, it may be easier today. For those who have not, we're going to try to make sense of it. But in order to make sense of it, we need to know what the structure of DNA is. Okay? And therefore, let's try to brainstorm out. This particular chapter, which GH3 topics are very relevant. The first one, I've already told you. Biomolecules, specifically revised nucleic acids. Okay, it will come in extremely useful. It's highly unlikely you cannot understand this topic without understanding the structure of DNA first as a baseline. Okay, so that's one aspect of it. Another thing you could revise is on cells. After all, this is on how cells divide. So an understanding about the structure of the cells will help also. Okay, so these two things are the foundation of GH3 topics that will help you understand this topic a lot better. So let's start first with biomolecules, nucleic acids. What is the structure of DNA? Okay, everyone got some time to buzz? Okay, so let's run through some of the structural points. Okay, the structure of DNA. Number one, Xavier.
It's a double helix structure.
Okay, good. It is a double helix structure. Double helix. The idea of double helix means that there are two strands. Yeah. I'm going to represent the two strands today on the whiteboard, not as a helix, but as just a ladder. Okay, just a ladder today. Double helix. But I'm just going to do the ladder, no helix. I think I'm going to instead take this okay, so we have a double helix on the whiteboard, but just represented by two strips. Okay, we take this as a ladder. Maybe this can jog back some memories. So thank you, Xavier. Double helix. Okay, anything else we can work with? Asher? Structure of DNA. Oh, okay. Hydrogen bonds. Where can you find the hydrogen bonds? Okay, between the base pair. So Asher talks about bonds. Okay, let's talk about bonds then. The bonds involve hydrogen bonds. And Asher said you can find them between the base pairs. So kind of here, this is where you will find the hydrogen bonds. Yeah, between the base pairs. Okay, let me just close that up. So that's what's holding the zipper together. Okay. Anything else? Jing complementary base pairing. Can we elaborate on that?
So on the bases, the A will point to T and C will point to G.
A will complementary base pair to T, C with G. Okay? Okay. Remember RNA? Yeah? RNA, if there's a slight difference. If I want to pair up, RNA doesn't have T, it has U instead. That's how the pairing will go. Okay, any more? Okay, in the complementary based pairing, right? Fundamentally, we work on hydrogen bonds also. Any more structures? One need? Anti-parallel. Okay. Will you elaborate on what anti-parallel means? The 3 end of one strand will bind to the 5 end of one of the other strand. Okay. So where's the 3? Where's the 5? Great question, right? So how do you recognize 3? How do you recognize 5? Okay, it has to do with carbon numbering, right? But if you don't understand how you number the carbons, how would you find out? Okay, the easiest way would be, let's look at nucleotide. If we look at nucleotide, the 3 hydroxyl group is always bonded to carbon number 3. Okay, hydroxyl group is bonded to carbon number 3. That makes it always a 3' end. The phosphate group is bonded to carbon number 5. That makes it the 5' end. So if we look at the entire strand over here, the 5' end is here. The 3' end is here. Kind of like what Wan Ling was saying, 5' with the 3' of the other strand. Anti-parallel. Notice that 1 goes in 5 to 3. And if I look directly below 3 to 5, that's the idea of anti-parallel. Okay, so a little bit more on the structure. Finally, maybe just one last question. When we form DNA, we find that to create a polynucleotide, we need many nucleotides. So if I were to put another nucleotide here, what is the bond holding them together? Phosphodiester bond. So that's the final one. If we talk about bonds, there are phosphodiester bonds that are found between nucleotides holding them together. They are a form of covalent bond. Okay, that is quite strong. Hydrogen bonds are the weaker ones. But by sheer number, it makes it quite strong. Okay, so we have phosphodiester bonds with covalent bonds. Finally, let's talk about the nucleotides. What's the difference between the nucleotides of DNA and RNA? Okay, let's do the sugar. The sugar is the difference. I did not show the difference in what I've made. I simply represented it by the RNA version is a darker version for today. Okay? But take note, if you really want to know the difference, if you go back and revise, it has to do with the idea that at the bottom right corner, one of them has OH, one of them doesn't. The one with OH belongs to RNA. The one without OH is the DNA one. It's what we call it deoxyribonucleotide. It doesn't have this oxy, the hydroxyl group. Okay? So the RNA is the one without OH. This one is the one, RNA is the one with OH, DNA is the one without OH. So if I want to be very specific, each one of this, it's not really a nucleotide, specifically it's a deoxyribonucleotide. Okay, each one is a deoxyribonucleotide. I think that is the foundation of the structure of DNA. It will come in really useful in understanding what's going to happen next. Okay, we'll take a little pause here for you to notate. For those that are ready, I'd like you to start reading the process of DNA replication. Okay, so meaning to say page 85, page 86, give it a quick read. Give it a quick read, underline portions that don't quite make sense. You could try this also. Try to identify all the enzymes used in this process. Everything that takes place in the body requires enzymes. Find out the enzymes involved in DNA replication and give them a highlight. Yes. Where? Where's the replication? Oh, Mr. Foster, good. The O is called the replication. For those that done, you can find a people partner. You just share with them the enzymes they've identified involved in this process. Technically, another GH3 topic would be enzymes. Not sure as you read whether visually you can imagine what's happening. Okay, talk to your friends. These are just words. Usually, can you imagine what's happening? Okay, I will start at 11:30. Maybe earlier than that. Okay, alright, let's begin. Let's try and make sense of DNA replication now. Okay, first and foremost, if we start on page 86, 86 is where the action starts to take place. Okay, on page 86. Okay, let's go through that. Proteins involved in the initiation of DNA replication recognize and bind to the origin of replication, causing the DNA double helix molecule to separate and open up to form single strands. What does that look like? Okay, so if we look at DNA, DNA is double helix. What the proteins do is that they unwind them. Okay, they unwind them. Rather, let me see that. See, the drawing I've made is not quite accurate. DNA is double-stranded, yeah? And we know that this double strand is also slightly twisted, right? And that's what forms a helix. Okay, so when they say they're going to unwind them at the origin of replication, what they mean to say, and then you read a few lines down, a replication bubble is formed. What they mean to say is this: at certain points, right, what the proteins can do is that they unwind them form a replication bubble. We call it a bubble because on a macroscopic scale, looks kind of like a bubble because they open up a small little segment such that now I have single strands over here. Okay, so now I've opened it up slightly. It's like I've opened up the zipper just at one small little segment. Now we call this a bubble, a replication bubble. This replication bubble is useful for the enzymes to go in to start replicating DNA. Right? So this is the starting point where replication of DNA begins. So the idea now is this. The enzymes that are going to come in next, they are going to create new DNA. I'm going to use red to represent a new DNA. They're going to create new DNA, new two single-stranded DNA, and they'll start synthesizing from there on. That's how we synthesize new DNA. In reality, there won't be just one replication bubble. There'll be many bubbles. So imagine a bubble just adjacent to it, and another bubble adjacent to it. To synthesize a total new two strands of DNA, we synthesize them in parts. When the bubbles meet, then it will connect to form an entire long strand of new DNA. So we work with many bubbles. Each bubble is like a small little factory where we use single strand of DNA as a template to create new DNA. When the bubbles come closer and closer to each other, like a merging bubble, then the new strands of DNA will connect and will form totally new strands. Okay? So many bubbles will form at any one time. But our focus for today is just at one bubble. We're just going to look at one bubble, how we synthesize new DNA. More specifically, I'm going to look at just one segment of the bubble, one half of the bubble. We're going to look at this portion over here. Okay, just going to look at this portion over here of the bubble. So I'm going to zoom in right now. I'm going to make the bubble right now. So imagine this is just one half of the bubble. Okay, the other half will go down this way. Then we have a bubble in the middle. Okay, so our focus is just one half of a bubble today. But perhaps this is where our story begins also. If you read your textbook, what's the first enzyme involved to create this bubble? Aptly name is called. It's called helicase or helicase. That's because its role is to unwind the helix to create the replication bubble. Yeah? Yeah? What?
We found a meme on this.
What is it? Wait, is it something with unzipping?
Yeah.
Okay, so we're now going to unzip this. Okay, so this is helicase. Its role is to unwind DNA. Okay, so what is not mentioned in the textbook but very subtly implied is also this. Therefore, what bonds are being broken here to unwind the DNA? Zhi? What bonds are being broken when helicase goes down and unzips this whole thing? Hydrogen bonds. It's implied. It's not explicitly said in your textbook, but it's implied over there. Okay, so let's unzip this fella. Did they explicitly say? Oh, oops. Okay. Okay, let's unzip this fella. Okay, helicase is going to go down. Can you help? Toju, help me unzip. The DNA. Okay, Toju. Yes. Okay, come up. Okay, as helicase goes through, help me unzip the DNA. Okay, but where is helicase at? Helicase is somewhere over here. Right? It's starting to create this bubble. Right? Okay, so I'm going to run down. I'm going to break the hydrogen bonds. Okay, ready? Let's go. Oh yeah. Okay, so we unzip a little bit of the DNA. Okay, hydrogen bonds have been broken. Okay, maybe just to make it a little bit easier for us to visualize, I'm going to move up a bit. Okay? Okay, so we've broken a lot of hydrogen bonds. That's the role of helicase. Function number one. Okay, that's the role of helicase. Okay, helicase job is done. Chongchu's job is done. Thanks, Chongchu. Okay, this is where things start to get a little bit complicated, but if we get it, it gets easier. What's the next enzyme involved? Yeah, what's the next enzyme involved? Primates. What's the function of primates? Okay, here I'm going to put primates. I'm going to provide RNA molecules. Sorry, ribonucleotides, not deoxyribonucleotides. And here I have primates. The beauty of it is this. Once I've unwound the DNA, each one can serve as a template for me to create new DNA. Okay, each one has a template. So this is a template. This can also serve as a template. Anyone daring enough to try? What's the textbook trying to show? What's the role of primates? How does it relate to the templates? How does it relate to the ribonucleotides that make up RNA over here? Okay, anyone daring enough to try? If not, I'm going to call someone whose name starts with D for daring. Oh, it's only dog. Dog? Daring dog? Okay, come dog. You hold your textbook with you if you want to. You'll try and make sense of what's happening. What's the role of primates? Do what you wish to. Okay, what's the role of primates? What does primates do? Primates. Sorry? Go ahead, do what you want.
How do you catch it? Like,
substitute, right? So you would want a complementary base pair with A. Okay?
Then the T that's fine. Put more.
Okay, Ken. Okay, thank you. What have you created here? What is this called? It's called primer. Okay, thank you, Tom. The primer has been created by this enzyme called primase. Okay, then let's talk a little bit about how DNA is synthesized. Even RNA. What Dom did was this. He added the RNA, and then he added the next ribonucleotide in to form a phosphodiester bond. Okay, I'd like you to think of one thing up. When we synthesize polynucleotides, we always synthesize them in 5 to 3 direction. Okay, when we make new RNA or DNA, it's always in this direction. What that means is this. If I were to look at a nucleotide, I can only add it this way. 5, 3, I always add it to the 3, I always add it to the 3, I always add it to the 3. 5 to 3 direction is the direction we take for synthesis. What Dom did, right, is also synthesis by the opposite way. 3 to 5, and then I add it to 5, and then I add it to the 5. Actually, in reality, we don't have the enzymes to do that. We only have the enzymes in our body to do from 5 to 3. So what you did is correct if we go in the opposite way. What if we did this? If we did this way, G, and then after that, a C, then after that, a G, then after that, A, and after that, a T. Okay? That is how you will synthesize something new. Okay, but we also have a problem now. Huh? If you read the textbook a little bit more, it says that the function of the primer is so that the next enzyme can come in to do its job. Okay, I would like everyone to ignore the bottom portion for now. Okay, it will be easier to understand if I work with the top template strand first. Okay, then you can start asking questions about how we're going to resolve this issue later on. Okay, so let's work with the one on the top. Here, T will go with U, right? A. Okay, T will go with A. And then A will go with U. Okay, so am I going to 5 to 3? I am. 5 to 3, I add it on to 3. I add it on to 3. Correct directionality, right? Primase has synthesized in primer. In reality, your textbook it says 8 to 10. But for the sake of the length of the whiteboard, I'm only going to work with primer with 3. Okay? So let's just focus on the top for now. We ignore the one on the bottom. We will come back to it later on as an extension. Where do we go from here? Which enzyme is involved next? DNA polymerase. Can we read carefully? What's the function of DNA polymerase? Okay, adds deoxyribonucleotides. Where? This is where I'll get the next person up to try. There's one more person whose name starts with D. Delphina, I just recall. Come Delphina, daring Delphina. Okay, Delphina will now try to synthesize the new DNA strand using DNA polymerase. How does it relate to the primer? Right, how does it relate to the primer? Or Delphina and Rachel can come up together also. You have to come up together. Right? You can't spell daring without a D and an R. Is there A in the daring? Who's A in the? Asher. Bracket Asher. As well as. Okay. Okay, maybe Asher, you go and help Delphina. So D and A. so big, right? It would be more cool, right? Yeah? Okay, so now we have DNA. I'm N. Wait, oh my God. Okay, yeah. Okay. So now let's synthesize a new strand, huh? Okay. Okay, so Asher and Delphina. Okay. How do we relate it all together? I have some deoxyribonucleotides here you can work with. What they've just done is kind of represent what was said in the textbook. That you kind of add deoxyribonucleotides to the primer. Did they add it the correct way? Yeah. They added the deoxyribonucleotide to the 3' OH group of the primer. They synthesized it in the 5' to 3' direction. Can you see the directionality? This is the 5', this is the 3'. If I describe the synthesis of the new DNA in the 5' to 3' direction, how do I describe the direction in which I read the template? It is in the opposite direction. So you read the template in the 3 to 5, but you synthesize the new DNA in the 5 to 3. Okay? This is how we use it in words. Okay? I read the template in 5 to 3, but I'm synthesizing new DNA in 3 to 5. I read the template in 3 to 5, and I'm synthesizing new one in 5 to 3. Yeah? I have so many words. Sorry, chalky over there. I think I confused your brain. Okay? Then a student from the next class asks, right? What does that mean? My new DNA is a mixture of DNA and RNA along the entire length. It's kind of odd now, right? But that's not the case. New DNA is totally DNA. It's not a mixture of RNA and DNA. But here we clearly see that there's some RNA and there's DNA. So the next enzyme comes into play. What's the next enzyme involved? Sorry? You don't know. You read carefully. What's the next enzyme involved? DNA ligase. If you read very carefully, there's another enzyme. Yeah. It's just called other DNA polymerase. So there are many DNA polymerases. This is another DNA polymerase. If you want to stretch yourself a bit, I want to find out the name, right? Actually, this DNA polymerase is called DNA polymerase alpha. This one is delta. There are many DNA polymerase. This one is the delta one, this is the alpha one. But in your writing, just say other DNA polymerase. Okay? So, let's make use of another DNA polymerase now. What's the function of this DNA polymerase? Its function is do this. It kind of swaps out the primer for deoxyribonucleotides. You see that in your textbook? It kind of swaps it out. So let's do a mutual swap. Let's swap up. So let's swap. A will swap with A. Okay. U will swap with T. C will swap with G. C. C will swap with C lah. What am I talking about? C will swap with C. Okay. So I've synthesized new DNA strand, a short one to replace the primer. That's the function of the other DNA polymerase. Okay. And what's the function of DNA ligase? Okay. The reality is this. Notice that, remember what we said? DNA polymerase can only add things in a 5 to 3 direction. Right? So technically, the it cannot add this C to this G over here. Think about it, right? You cannot add this C, I cannot connect it this way. This is just not the role of DNA polymerase. You can only add in a 5 to 3. To connect this to here, I'm actually adding it to the 5 side. Right? That's not the role of DNA polymerase. You cannot do that. So actually right now, I have a slight break. I've only connected these 3, but not quite the last part. Yeah? But that's the role of DNA ligase. DNA ligase just seals off all the missing links. DNA ligase can do that. That's its last role, is to seal up the missing links. The missing link is just between the swapped out primer and the new strand. Okay, that's the role of DNA ligase. That's why they say its role is to kind of synthesize phosphodiester bonds. Once you understand the basics, a lot of questions will start to pop up in your mind. That you start to question a bit more. Okay, but I just want you to know the basics for now. The further questions, we can take it. Okay, we still have time. Okay, so imagine this, we repeat this at the bottom strand, and we repeat this along the entire length of DNA. We will eventually get 2 new strands of DNA, right? That's DNA replication. This is a simple way of understanding this. The process is a little bit more complicated if you look at this carefully. Okay, but if you understand whatever we just went through, it's good enough. If you want to push yourself a bit more, you realize the dilemma we have. We go back to Dom's primer that we made just now. Let's put Dom's primer over here. Is there a problem? Remember the rule? We synthesize in the 5 in 3 direction. Dom's primer over here, how do we do anything with it? Maybe I take away some, then we can think about it. Okay, so from here onwards is extra knowledge. Okay, but if you want to think about it, it's quite interesting. So how? We cannot add new DNA this way, right? Right now, this is a 5' end. Yeah, so we cannot add more. How?
But you can add ribonucleotides that way.
Where?
You can add the ribonucleotides that way.
Which way?
Like 5 to 3.
So, are you saying this way?
That way.
Or add it this way?
I mean your how is your ribonucleotide added?
Oh, okay, so you're saying perhaps we put it here instead, is it?
No, I'm just genuinely asking a question. Like how is the ribonucleotide added? In what direction? Is there a directionality?
Ribonucleotides do have a directionality, it's also 5 to 3. It's also 5 to 3. But perhaps we brought an interesting point. Must you put the primer here? Could you put the primer here, like further up? Would that solve the problem a little bit? Because if I put the primer slightly closer to the where it's unzipping, right? Then now I can add new DNA like that. Yeah, 5 to 3, right? So we can solve the issue a little bit this way, right? But what if helicase comes in? Where's my helicase? What if helicase comes in? We unzip some more. Zip some more, unzip some more. Remember it's endless, I unzip some more. Now this portion here becomes irrelevant. The top strand is still okay, notice? I can still actually, if the primer is here, I can continue going on endlessly, right? But the bottom strand, notice, I kind of get stuck if I want to follow on the zipper, right? Yeah, so this is a problem, but the body has found a way to solve this problem. You want to speculate how?
Another bubble.
Or you're going to read up about it. Yeah? Okay, why the top here, bottom cannot? You see, I put the primer here. Where's my primer? I put the primer here. A, U, and C. is 5 to 3, right? Yeah, I can just keep adding on. This one is 3 to 5. I cannot add it on this way, even though my enzyme helicase is zipping, unzipping this way. I can only add it in this direction. It'd be weird, right? Okay, then, can you just notice actually the same problem on top? On top, remember helicase is unzipping this way, but on the other end, there's another helicase unzipping that way. If you look at the strand on top, technically I cannot write anything in this direction, right? As it's unzipping this way. So you have a problem. Yeah, the helicase, each one take one side and then unzip, unzip, unzip. Then the bubbles merge into bigger bubbles. Yeah? So you notice, actually in both directions, one strand, this one I can synthesize in the continuous fashion. This one, let's say I shift it up, right? I can synthesize here, but then I cannot solve it here, right? The same thing happens on top. I actually cannot synthesize anything in this direction, even though helicase is also going in that direction. So how do we solve this problem? Maybe another bubble can come and join, then we solve the issue. Sushi? You felt like you were getting somewhere?
Like because all the bubbles are like next to each other, right?
Yeah.
So like over there where the bubble is, right?
Yeah.
Like on the right hand side, like over there, right? So like the helicase is going this way, but then bubbles from next to this bubble is going to this. Like wait, actually I don't know what.
But it's an interesting thought. I also have not explored that thought before. Yeah? I will not tell you the answer. I will not tell you the answer. You can find the answer very easily if you go and read up more. But it's very interesting. The process is a little bit more complicated than what we presented to you. Okay? The key thing, if you want to go and read up more to make sense of how we solve this issue, the key word will be Okazaki fragments. If you go and Google this, things will become clearer for you. If you are interested, yeah? It's not. Okay, Okazaki fragments is not. But I just wanted to stretch you a little bit more. Because if you question it a little bit more, you realize what I just presented to you, that it doesn't run very smoothly. Okay? If you want to find out more, Okazaki fragments is what you can Google to find out more. However, if you are satisfied with just understanding how I synthesize one continuous strand on top, that's actually sufficient for our level. You go on to the JC side, you will learn about how we solve this problem. But I'm telling you, this problem is you can understand this problem and how it is solved if you just go and read it. Okay? Then maybe next time around when we come back to class, I'll ask someone to share how this problem is solved. Right, then we can all learn a little bit more. Or you can think a little bit more and think how we might solve this problem. Okay? Okay, so that's all for today. This is the story of how DNA is replicated. Okay? But there's one thing we could go and explore a lot more, is how we solve this issue of this obstruction, because we can only synthesize things 5 to 3, 5. Okay, if you like to, you could spend some time looking at the internet now to read about Okazaki fragments. Okay, then maybe we'll share a little bit next time round, the next lesson, and then you'll see how things fall into place. Yes. Why not just put DNA? Okay. Because you don't have the it sounds very lame, I always use this excuse, but it's true. Actually don't have the enzyme to put DNA deoxyribonucleotide first. Yeah. But we do have an enzyme that can put RNA immediately. And this is how we synthesize mRNA from scratch. So we are kind of using enzymes that can do mRNA, but we repurpose it. Yeah. So we can attach RNA from scratch, but not DNA. Yeah. And we see that in how we synthesize mRNA. Okay, Teja asked a question. how come we must add RNA primer? Why not we add a short segment of DNA, then we use it to build more DNA? The layman's answer I can give you, but it's the truth, is that we don't have the enzymes to put DNA first. We have the enzymes to attach RNA. The second reason is that actually this serves as a checkpoint for the cell. The cell uses all these primers as a guide to tell them when new DNA has been synthesized. So if an enzyme comes by and it looks at, oh, here's a primer and here's new DNA, I better check that whatever has been newly synthesized is correctly paired. If it's correctly paired, then I swap it out. So actually all these primers serves as a guide for the cell to tell where new DNA has been synthesized for me to do a double check. That's its secondary function. Okay? Okay, you can spend some time researching about Okazaki fragments. And we can talk about it a lot more in the next lesson. This is FYI. You will learn it in the JC side. But it's an interesting problem that we can solve. Okay, so for our next lesson, if you want to prepare for it, what you can do is to read up on the Messelson and Stahl experiment on page 87. This is the toughest part for the synthesis phase of interface. Students often find this one page the killer of this topic. Okay, you can read up more about that to prep for the next lesson. David, thank you. You can stop the recording.